Hi Priyadarshi.
Yeah.
You're all set?
Yeah, I'm set. So it's not until 3 o'clock, so we wait.
Okay. I'm ready. So whenever. So by the way, Vikram, you're there?
Yes, yes.
So my slides are visible, right?
Yeah.
Just
roll
it forward and backward.
Yeah, yeah. Okay. Hold on.
I can see your face in the slides.
Okay. Yeah. Did the slide move now?
Yeah, it's moving.
Okay. It's moving, right?
Cursor.
Yeah, it's moving.
Show the cursor.
Yeah, but I'm not able to
cursor
see.
is also showing. Yeah, fine.
No, hold on. I stopped presenting.
Yeah. So
Sreedhar.
yeah.
Switch on your lights. All the lights.
All lights they're on. My lights are like this. Both of the lights are on.
My lights are okay. Hi Alpana. How are you?
Hi, good, doing well.
Yeah, I don't know why my lights are dimmer than usual.
We are live, huh? This meeting is live.
Okay, so we start in 10 minutes?
Yeah, sharp at 3.
Okay.
Yeah.
Good afternoon, everyone. I think we'll start. Because we are live, right? Good afternoon, everyone. Welcome to our next round of Science Setu seminar. So these seminars are being organized as a part of is the initiative of the Government of India to celebrate and commemorate 75 years of Indian independence. And as you might know, these events have started since March this year and will until August 2023. So with this brief introduction of the event, I'll request Dr. Priyadarshi Basu to please introduce our speaker for today.
Thank you, Alpana. It gives me immense pleasure to introduce Dr. Sreedhar Chinnaswamy, my colleague at National Institute of Biomedical Genomics. Dr. Chinnaswamy graduated as a veterinary clinician at the Bangalore Veterinary College, Bangalore, in 1999. Then his interest in basic sciences drew him to do a master's degree in animal biochemistry from the Indian Veterinary Research Institute, Izzat Nagar. From there, he went to the New Mexico State University, Locus in 2003 and obtained a research MS degree in chemistry from the Department of Chemistry and Biochemistry in 2005. He worked on the Hepatitis C Virus core protein for his thesis. From NMSU, he went to Texas A&M University, USA in 2005 and worked to understand the requirements of the HCV RNA-dependent RNA polymerase for de novo RNA synthesis. Dr. Chinnaswamy's work showed for the first time that a flexible loop on the surface of the protein called the delta-1 loop that linked the fingers and thumb domains of the RNA-dependent RNA polymerase was responsible for a locking mechanism that could elegantly explain the different modes of RNA synthesis by the polymerase. Dr. Chinnaswamy, after completing his PhD, came back to India to take up a faculty position at the National Institute of Biomedical Genomics in Kolani, India, and has since focused his work on the role of type 3 interference in the immunity against RNA viruses, including HCV. He was awarded the Wellcome Trust DBT Intermediate Fellowship in 2018 to investigate the genetics and functions involving the newest member of the type 3 interferon family, INF-L4. Today, he will speak about the work he has carried out at NIBMG in the last few years. So, Dr. Chinnaswamy, over to you. Thank you.
Thank you, Priyadarshi. Can you hear me and see my first slide?
Yes, yes.
Okay, thank you. Okay, I hope many people have joined this event. I welcome you to my talk, and again, thanks to my colleague, Dr. Priyadarshi Basu, for the introduction. So today, I'll be talking about the type 3 interferon locus in humans, because there are certain interesting genetic variants present at this locus, which have very high significance in human health and disease. So this talk is going to be a little more focused than the talks that you have heard so far from my colleagues at NIBMG. As the title itself says, it's about the interferon lambda, which is also called the type 3 interferon genetic variants, and how they are associated with human not only infectious disease but also inflammatory diseases. Now, the interferon lambda locus present on chromosome 19 in humans comprise basically of 4 genes, as shown in the bottom here: IFNL1, 2, 3, and 4. These are all large extent homologous genes, suggesting that they are all evolved from gene duplication events. Now, IFNL1, 2, 3 were discovered after the major thrust in the human genomic in the genomics revolution, which was the elucidation of the Human Genome Project, which was published in 2000. So following that, in the year 2003, IFNL1, 2, 3 were discovered. And IFNL4 was discovered much more recently, that is in 2013. Incidentally, after another major event that happened in genomics revolution, which is the which is the advent and widespread use of the Genome-Wide Association study, which opened up new avenues in studying of complex diseases. So I will go through these things in a little more detail in the coming slides. But until 2000, although the IFNL 1, 2, 3, the locus itself was discovered in 2003, until 2009, when the Genome-Wide Association studies happened in hepatitis C virus infections. This locus remained mostly of academic importance. So, but after the 2009 GWAS, as the Genome-Wide Association study is pronounced in short, after the HCV hepatitis C virus GWAS, this locus has really come into the limelight, and researchers across the world, immunologists specifically, have become very interested in this locus and have been carrying out phenomenal work in different aspects, including genetics, biology, and everything has happened after 2009, after the GWAS results. Since the GWAS I mentioned happened in hepatitis C virus infection, so let me briefly take you through hepatitis C virus infection and how the GWAS came through. So hepatitis C virus is another positive strand RNA virus like the COVID virus, but it belongs to a different family, and it has much smaller genome, about 3 times smaller genome than the COVID virus that we are very familiar with by now. Unlike the COVID virus, it causes chronic infections of the liver, leading to cirrhosis and even cancer. A lot of people around the world are affected by this virus, including many Indians are supposedly carrying this virus. There is no vaccine for this virus as well. Until 2013-14, the treatment of choice was what is called as pegylated interferon. It is an interferon alpha, which is pegylated, and in combination with ribavirin, which is a nucleotide analog. Ribavirin mimics GTP, which is used to build DNA in our normal cells. And therefore, the interferon, which is an antiviral cytokine, along with this GTP analog, which gets incorporated into the viral genome during replication, but it prevents the further elongation of the polymerase reaction. Therefore, this was used as a drug to prevent HCV replication. So until 2013-14, this was the treatment of choice. But after that, much effective drugs called as direct acting antivirals have come into the practice, and there is phenomenal success that has happened due to these. And you all know, last year's Nobel went to the discovery in hepatitis C virus to 3 eminent scientists across the world. So that is this. But until 2013-14, the treatment of choice was peg-interferon alpha ribavirin, which was effective only in a fraction of people. For example, there are about 7 genotypes of the hepatitis C virus. If you are infected with genotype 1 of the virus, the chance of responding to treatment after the interferon ribavirin therapy was only about 50%. Anyway, so HCV gets transmitted through the blood or sexual contact, unprotected sexual contact, or any mechanism that will put the virus into the blood. So, as I said, let's say in genotype 1 infection, about 20% of the individuals can spontaneously clear the virus on their own. The remaining 80% go on to have the chronic infection, which is characterized by hepatitis, cirrhosis, and even HCC, which is hepatocellular carcinoma. So, and these 80% people, as I told, if they are given the interferon alpha ribavirin therapy, only 50% of them will respond to the treatment, others do not. Now, if you see the infection rate in different ethnicities, ethnicities are closely linked to genetics, right? Different ethnicities have originated from different sources, and therefore they carry a different distinct set of genomes. With variation built in within the ethnicities. Ethnicity is intricately linked to the genetic variation. And you see that the hepatitis C virus infection rate differs significantly across different ethnicities. For example, white, black, Hispanics, Asians, and Pacific Islanders. This is one example. This the graph on the right that is shown, which means that HCV infection has a big role that has to do with the genetics of the individual. And therefore, this became a very good candidate to carry out the genome-wide association studies, which are large-scale studies designed to determine what variation genetically is responsible for different phenotypes in two groups of individuals. Now, interferons are basically three classes: type 1, type 2, and type 3. Type 1 is the more famous ones because they were discovered much earlier and a lot of work has been done. There are about 5 of them. Interferon alpha has different isoforms. Type 2 interferons is characterized by a single interferon, interferon gamma, which is produced by mainly immune cells. The type 1 interferons are essentially produced by all nucleated cells and also can act on a lot of cells in humans. Whereas type 3 interferons, as I said, there are 4, but what is unique is that they can act on only a set of cells and tissues, mainly the epithelium. Epithelium, by the way, constitutes a large surface area. As you know, we have the respiratory tract, the digestive tract, the reproductive tract, and the skin is also epithelium origin. And also several other organs have cell types that have originated from epithelium. So type 3 interferons have receptors in all of these, and also some of the immune cells. Now, as I said, the Genome-Wide Association studies of 2009, which are the 3 important studies, they're almost very famous now, even after 10 years around. One of the successes of the genomics revolution, you could say, identified the interferon lambda locus, as I said, and they identified what are called as the single nucleotide polymorphisms, which go by the names like, for example, the one shown here, RS1297980, is a single nucleotide polymorphism. So, but let me introduce you a little bit on what is GWAS. I'm sure my colleagues in their previous talks have talked about what GWAS is, but for this presentation's sake, I'll give a little bit of an introduction. For example, this group of individuals, let's say they responded to treatment. Although the figure says it's the heart disease, but we can change it to HCV treatment response. And this group of individuals did not respond to HCV treatment response. Now, obviously, if there is a genetic cause for this differential response, this genetic variation has to be identified. Now, one form of genetic variation that you see is in the form of what are called as single nucleotide polymorphisms, or SNPs. That is, at a one nucleotide, you find one allele, that is, let's say, T in one individual, but in another individual, like for example, I might have T, you might have G at a particular nucleotide position in our genome. Now, GWAS is a method where you look at such variation across the genome, all the chromosomes, as much as the technology allows us to do. For example, there are more than 20 million SNPs or even more that may be present on the human genome. The technology allows us a limited capacity. About 5 million SNPs can be genotyped, meaning we can find out and go into these 5 million regions in our genome and say whether I have a C and you have a C or a T. So that is what it is. So when you do this in these cases, or the people who responded to treatment and those who did not respond to treatment all across the genome, and see what is the variation and perform a statistical test to see against each SNP position, if you find a particular variation to be very prominently present, for example, a large number of people who responded to treatment has G at a particular SNP position, and similarly a large number of people who did not respond to treatment have an alternate nucleotide, for example, alternate allele, you would say, let's say T. So G in one, T in another group, and if it's statistically different, statistically significant, then you say that is a GWAS hit. That means it is highly likely that this variation originating from this position in the genome is likely leading to a different response when you take the same drug. So that is the basis of a GWAS result. And GWAS results are usually shown by what is called as a Manhattan plot, that is shown here. So basically every chromosome is shown which with the highest significantly associated SNP coming up as a tall building here. If you see RS1297980, I'll just take the last 3 digits from now on. So RS860 was found to be the strongest hit that came out. That was different in individuals who responded to treatment and people who did not respond to treatment. So this is RS860 here. This was very close to the IFNL3. It was also called as IL28B at that time. So it was very close to IFNL3. Another SNP, RS917, was also a strong hit in the GWAS in some populations, but that is also about 7 kb away from the IFNL3. Essentially saying that this locus seems to be very important in how a person responds to treatment to hepatitis C virus infection. So, but now the problem with GWAS is usually, the association is by proxy. What do we mean by that? As I said, there are more than 20 million SNPs that could be present on the human genome, and it's technically impossible for us to get all of these SNPs in what is called as a GWAS chip, which is basically a microarray that will contain probes that will tell you whether your genome has a G or a C at different SNP positions. So the company who manufacture these chips get around this problem of dealing with large number of SNPs by utilizing a phenomenon called as linkage disequilibrium, or LD, that's shown here in red. So what is LD? LD is a phenomenon by which contiguous regions of DNA, large stretches of DNA it could be, that get transmitted to successive generations without getting crossed over. As you know, in meiosis, the chromosomes get crossed over, but in what is called as an LD block, the crossover chance is very less, and therefore the entire block gets transmitted from one generation to another as one single unit. So what is the significance of this? For example, if there are 100 SNPs within this LD block, if you know the information about one SNP, you basically can know the information of the remaining 99 SNPs because they all go together. And therefore, by picking one SNP in that LD block, you are essentially getting information of the entire thing. So the companies just pick these LD blocks, which are specific to specific populations, and pick a few SNPs from those LD blocks and populate their GWAS chips. And then you do the study and you get the hits. So for example, we got RS860 as the strongest hit in the HCV GWAS. Now, SNP is one, is kind of variant. A causal variant is a different SNP. A causal SNP is a single nucleotide polymorphism which will change at least either the expression or the function of a nearby gene. What do I mean by that? For example, if 860, if it can be considered as a causal variant, it should either change the expression of a nearby gene, in this case IFNL3, it should either increase or decrease the expression, or it should cause an amino acid change. For that, it has to be within the gene. So obviously this one is not within the gene, therefore it cannot function in a way that it will change an amino acid in the protein and change its function. So these are the two minimal mechanisms by which an SNP can be changing the phenotype, changing the biology basically. So when you do a GWAS and come up with the strongest hit, it need not necessarily the SNP could be representing the causal variant, which is shown in the red here. Because of the LD phenomenon, and the companies pick only a few representative SNPs, proxy SNPs in that LD block. So for example, they might have picked the yellow one, and that shows the highest hit, highest significance, because it is very closely linked to the causal variant. It is not showing the significance because it is causing the phenotype. It is causing the phenotype. It is not causing this phenotype, but it has come up in the test because it is in close relation with the actual causes. So the challenge after GWAS becomes how to identify this red variant, which is actually the causal variant. There are several techniques to do that, and one interesting study that came up in 2011 was called as recombinant mapping, came from a group in Italy. They did a very fantastic analysis in HCV patients, and they predicted that 4 SNPs, as shown here, could be the causal variants. Now, by the time I started my work, other groups had already shown that RS142, which actually causes an amino acid change in the gene, this is IFNL3 gene exon, And 219, RS219, which is present in the promoter of IFNL3, these do not have any functional significance, meaning they could also be proxy SNPs. They were just getting associated because of the closeness to some other causal variant. So we are left with RS217, RS813. So in 2013, our group was the first to identify and show that RS813, which is present in the 5' UTR. By the way, this is present. 813 is present in 5' UTR of IFNL3. 217 is present in 3' UTR of IFNL3. So this 813, which is very close to the promoter of IFNL3, was just one nucleotide away from a NF-kappa B binding site. The binding site shown here. So NF-kappa B is, as you know, is a transcription factor which is very important in the transcription of several immunity-related genes. So this SNP being very close to an NF-kappa B binding site, we predicted that this might be affecting the binding of this transcription factor, and therefore it could be affecting the expression of IFNL3. And we show experimental evidence for that. But just for a while, I'd like to change gears here. Another group from University of Washington and Seattle showed the same year that the other variant RS217 present in the 3' UTR also was a causal variant because it interfered with the microRNA binding site, and therefore you know microRNA will regulate translation of mRNAs, and therefore that was affected by this SNP. And also it was interfering by what is called as the AU-rich element mediated decay pathway. As you see, the 3' UTR has 3 AU-rich elements here. The 217, the alternate alleles were both interfering with the AMD pathway, as well as they were changing the secondary structure, changing the stability of the RNA. In summary, what they found was that the protective allele, meaning the people who showed a positive response to HCV treatment, were the G allele carriers, their mRNA, IFNL3 mRNA levels were much more stable compared to the ones who did not respond to the treatment. They are also called as the major allele and the minor allele. Major allele means it depends on the population, but usually apart from the African population, the major alleles of the remaining populations are the ones which are found in the higher frequency, and the minor allele is the one which is found in lower frequency. Now, in 2013, another phenomenal study showed that there was a new type 3 interferon just upstream of IFNL3. In fact, in the distal promoter of IFNL3, a new type 3 interferon was present called IFNL4. Now, this is an interesting interferon because it is generated out of a polymorphism. So it is not an SNP, not a single nucleotide variant, but it is a double nucleotide variant, or because it has two alleles that are changed. Either you have delta G or the TT alleles. So delta means one TT is gone, the G replaces the other T. So the two alleles are, so this is a dinucleotide variant, TT and delta G. So people who have the delta G allele at this position that goes by this RS number will open up an open reading frame for the fully functional interferon lambda 4 protein. People with the TT allele will abolish the open reading frame, and they will not produce interferon lambda 4. And therefore, it could be that you could have the delta G allele where you will have a new type 3 interferon that you can produce. Or if you have the TT allele, you don't have this new type 3 interferon. So that's what that was it. So in summary, I have told that there are at least two sets of genetic variants present at this locus. Two variants I've already told, RS217, RS813, which will control the expression, either transcription or translational because of mRNA stability of IFNL3. This is one set. The other set is IFNL4, the dinucleotide variant that will either create the new type 3 interferon IFNL4 gene or not. But the problem when you associate with this locus with any disease, for example, if you see variation in this locus, any of the three variants that I've shown here, and you see an association with a particular disease, let us say hepatitis C virus treatment response, Now the question arises whether it is because of IFNL3 expression changes is the phenotype happening, or because there is a new type 3 interferon lambda 4 that is causing the effect. You could use statistical methods if they are farther apart and they are not linked. But unfortunately, in majority of world populations, they are closely linked and they are in high linkage disequilibrium. Therefore, using methods statistically, it is difficult to tease out which of the variant and therefore which of the interferon lambda G is responsible for the phenotype. So that is the challenge in the area, and my work has contributed to understanding this problem and probably to provide a solution to that. So as I said, the 813 is very close to the NF-kappa B binding site, which is shown in black here. The SNP site is shown here GC. As you see, there is only one nucleotide separating. So we hypothesize that this may be binding the alternate alleles G and C might be differentially binding to NF-kappa B. This is a co-crystal structure. P50 is a component of NF-kappa B along with the DNA and the P65. So what we did was we produced the P50 component, that is the one that's shown in yellow, and did what is called as an electrophoretic mobility shift assay. We took this DNA with either G or C and added this DNA, we labeled the DNA and then added this protein to interact with the DNA and ran it on native gels. That means if they are interacting, they are not disrupted, as opposed to they are run in SDS page. So we are not use SDS. This is a native gel. They can run in the gel. Either the free probe you could see like this, or if it is bound to the protein, it gets shifted. That's why it's called as a shift assay. So then you could see if your C allele is binding more or G allele is binding more. As you clearly see here, the C allele was binding more of P50 compared to the G allele. That means what we predicted was correct. That is, it may be affecting the binding of NF-kappa B, and therefore, if you do a reporter assay, we clearly see that the protective allele, that is the allele that is present in higher frequency in people who respond to HCV treatment, were showing higher gene expression compared to people who are carrying the non-protective allele. Now, in a recent work, we combined two of these SNPs, the 3' UTR and the 5' UTR SNPs, in the same assay and measured the luciferase or the reporter gene activity. And we found an interesting result where we see that only if you have both the non-protective, here non-protective is also the minor allele, which is present in the lower frequency. The non-protective allele, if both are present in both the SNP 5' and 3' UTR positions, you get a much reduced gene expression compared to either of the minor alleles in either of the SNPs carrying the respective minor alleles. So in other words, the non-protective effect you will see if the minor alleles are present at both 3' UTR and 5' UTR. So this is the story of the two causal variants at lambda 3. Now it is very difficult to find out causal variants after a GWAS, but in this case success was seen pretty early, and people were excited that we have found the causal variants. So the explanation could be that people with the protective alleles, because it increases IFNL3 expression, and therefore they are able to clear the virus when you provide the treatment, as opposed to people who have the minor alleles which do not, which have lower levels of IFNL3, and therefore somehow this lower level of IFNL3 is preventing the clearance of the infection. So problem solved, but there was actually another problem that came from the HCV clinics. So in hepatitis C virus clinics, it was very common knowledge. So what does an interferon do? An interferon usually goes out of the cell and binds to its receptors and activates the JAK-STAT pathway and increases the expression of what are called as ISGs, or interferon stimulated genes. There are more than 1,000 genes in this class, and that is how any interferon acts to get rid of a virus. So in HCV clinics, if you take a liver biopsy from a patient before treatment, before subjecting this patient to treatment, and do an RNA-seq, for example, to see how many ISGs are present, or an array experiment to see what is the expression of ISGs, it was known that people who responded as shown by R here and people who don't respond to treatment. So this is before getting the treatment. You take the liver biopsies of all the patients and then give them the treatment, and then some people respond, some people don't respond. Now you see what was their ISGs before treatment. It was common knowledge that people who eventually do not respond to treatment, the NR group, usually had a higher ISG. More red is more expression of ISG. So they had more expression of ISG, but somehow they were not clearing the virus. This is actually called as the Interferon Hcv paradox. So even though interferon activity is more in the livers of patients, that they cannot clear the virus, as opposed to patients who have lesser ISG expression, when they are given the interferon alpha ribavirin treatment, they respond better. Therefore, the hypothesis that IFNL3 increasing expression and clearing the virus may be wrong. So there should be some mechanism by which a minor allele carrier is actually giving rise to more ISG Gs in the liver because of some kind of interferon activity. And this was exactly found by a group in NIH in 2013, where they see that the delta G allele carriers, which is the minor allele carriers, are actually giving rise to a new interferon, type 3 interferon called as interferon lambda 4. And therefore, this group of individuals, the minor allele or non-protective allele carriers, they express a new interferon, which is interferon lambda 4, which gives rise to high amount of ISGs. And therefore, when you give a treatment with another interferon called interferon alpha, it will not further upregulate ISGs, which is required to kill the virus. As opposed to the TT carriers, where ISG is low, you give another interferon in the form of interferon alpha, it will immediately jack up the ISGs and kill the virus. So the mechanism was explained, and now everyone understood, okay, the people who do not respond are the ones who have this extra interferon. That means having this extra interferon was bad, at least as far as HCV is considered, and this is called as the paradox. This study also showed another interesting non-synonymous variant within IFNL4 at the 70th position. So there was a proline variant with the wild type activity, and there was a SNP present which changed the proline to serine, and that affected the activity of the lambda 4. The serine variant had lower activity. In other words, it has lower ISG stimulation. So remember, higher activity of this interferon is bad. Therefore, lower activity should be good. So I will come to what is the significance of this in a while. So now again people got excited. Okay, we got the answer. It is the interferon lambda 4. That is the mechanism of why some people do not respond to treatment in HCV infections. But some studies showed that IFNL4 has very poor transcription and translational abilities, meaning it is not highly expressed in cells. For example, it also has what is called as intron retention expression. For example, in mRNA, you should have only exons after splicing. But if you induce for interferon lambda 4, you see what are called as introns. Some of the introns get retained in the mRNA, which will not form a functional protein, and it will be degraded. So for example, if you in a common cell line like HUH7, if you induce for the type 3 interferons, you see very high expression of L3, lambda 3, but and also interferon lambda 4 intron retention form, IR form, which is inactive. But the active form, which is P179, is not at all produced. So people thought that how can this be when the protein is not even expressed? There is a problem in expression. They also showed that it has problems in loading to the ribosomes. This mRNA has problem in loading to the ribosomes, and therefore that is further preventing the translation of this mRNA, and therefore raising question marks on whether lambda 4 is expressed or not. If that is the case, how can this be the causal mechanism? So now after that, these variants became associated with number of the disease and health phenotypes. Some of them I have listed here, including type 2 diabetes, so several autoimmune conditions, even cancer, allergy, asthma, diseases like non-alcoholic fatty liver disease, and so on and so forth. Some of them I will name. So therefore, it becomes important for us to understand which is the causal mechanism. Is it lambda 3 or lambda 4? So in summary, what I have told so far is, if you are the major allele carriers, that means if both of your chromosomes have the major alleles, you express more lambda 3. There is no lambda 4 in you, and and this expresses some set of ISGs, interferon stimulated genes. And in HCV, if you get HCV, you have a protective phenotype. If you carry one copy of the minor allele, so one copy of the major allele and one copy of minor allele, or both of them are minor alleles. In that case, you do express lambda 3 because lambda 3 the expression itself is not under the control of polymorphism like lambda 4. You do express lambda 3, but you also express lambda 4. But your lambda 3 will be less compared to the major allele carriers. But you have an extra lambda for the lambda 4. And they will give almost similar set of ISGs, but very small differences. But paradoxically, that becomes a risk phenotype for you if you have a risk factor for you if you have hepatitis C virus infection. So using this, we did a small study using patients from India who had hepatitis C virus infections who had undergone treatment. And after treatment, they were classified either as responders to treatment, that is, they got rid of the virus, 52 of them, and 21 of them did not respond. That means they still carried the virus with them even after treatment with interferon alpha ribavirin. Now, the simple test would be you look at the polymorphism frequency of the alleles at the variant that gives rise to interferon lambda 4, that is RS815. But surprisingly, we saw that this did not associate with treatment response. So how do I say that? The p-value here is above 0.05, and therefore there is no association. Above p is 0.05 is not significant. That means lambda 4 generating variant did not associate with treatment response, which was a surprise to us, which probably suggests that it may not be the causal variant, right? Because the hypothesis is that, which is also a fact, is that a causal variant should show the best strength and significance of association with the phenotype. If the variant is causal, that should show, provided there are no other confounders, that should show the best strength and significance of association with the phenotype, which did not happen for this 815, and therefore raising questions on the mechanism involving lambda 4 as the causal mechanism. But surprisingly, we got strong association with another nearby SNP called as RS221. Now RS221 was actually, so it is present within the first exon of lambda 4. So this is the structure of the lambda 4 gene, 5 exons present in the distal promoter of interferon lambda 3. But interestingly, the CpG island connected to interferon lambda 3 largely overlaps with the interferon. The CpG island is the green rectangle shown here. It is overlapping with the interferon lambda 4 gene. So as I told, 221 is present in the exon. Now RS221 was not new because in 2011, a very well performed Australian study in a large group of patients using next generation sequencing technology had already identified 221 as the best predictor, and also they had predicted that this could be the causal variant because it is lying in the CpG island, and therefore it is somehow changing the nature of the CpG methylation in this region, and therefore this may be controlling the expression of interferon lambda 3. So we tested that hypothesis because we also got RS221 as the most best predicting SNP. And using some in vitro methods, we saw no change in activity with the wild type and the mutant alleles as far as RS221 was concerned. That means RS221 could not be doing its function by changing expression of IFNL3. There should be some other explanation to that. Now, using the RS444 SNP, I told you the 2013 NIH study identified another non-synonymous variant at the 70th position, which changed the function of the lambda 4. Now there are two variants we are talking about at lambda 4. One that actually gives rise to interferon lambda 4, that is RS815, this one, and another one which changes its activity and therefore its function. Therefore, how these two are connected to each other will decide whether, for example, if the wild type if you have the wild type allele that is present at the P70 position, then you get a high activity of lambda 4, which paradoxically cannot kill the virus, right? Now, if you have the low activity producing serine variant at lambda 4, therefore you will have a lesser activity of interferon lambda 4 that will give you a protective phenotype. So therefore, you should not only look at the variant that generates lambda 4, you should also look at the variant that is controlling its function. Therefore, the linking of these two things will decide the final phenotype. In other words, the RS444, which is changing the activity of interferon lambda 4, could be acting as a confounder. Now, what is a confounder? In simple terms, we'll take a very simple example here. For example, in beaches, when ice cream sales go up, shark attacks also go up, and therefore, if you do a test, it will be correlated. If you increase, if you just compare ice cream sales and shark attacks, they will go hand in hand, giving us the spurious association or misinterpretations saying that ice cream have something to do with shark attacks. But in essence, it is the heat wave that is doing this, because heat wave is being a confounder, is connected to both the independent variable, which is ice cream sales, as well as the dependent variable, which is shark attacks. So when heat wave goes up, people eat more ice creams, and the sharks are also out, and you get more shark attacks. And therefore, understanding this confounding effect becomes important in association studies. Now, a positive confounder will make your estimate go away from the null hypothesis. For example, if you hypothesize here 815, the variant that generates interferon lambda 4 is associated with the treatment response, your null hypothesis will be there is no association, right? But if there is a confounder, positive confounder, it will push this estimate away from the null hypothesis, suggesting that you will start to see an association even though there is no association, because it's a confounder that is connecting both your dependent as well as the independent variables and actually confusing your results. So it is better to, it is always important to understand that. We did a lot of experiments and we understood that RS221, the SNP that gave us the best significance was actually being confounded by RS444, because the way this the gene structure was in that region, the way the 444 was linked to 815 was ultimately linked to 221, suggested that 221 was always connected to the better phenotype of lambda 4 or the better form of lambda 4. That is the protective allele which generates the lower activity of lambda 4 was always associated with the RS221 to the major allele, and therefore when you do a test, the major allele, if it is connected to the protective phenotype, it starts to show a better effect compared to an SNP that has the opposite connection. Anyway, to cut the long story short, we established that 221 was actually not the causal variant, but it was actually getting a positive confounding effect because of something that was happening with the lambda 4 function and expression, and therefore providing indirect evidence that it indeed interferon lambda 4 is the causal variant. Now using statistical method, if you correct for the activity of for the RS44 variant, which changes its activity, now you see significance with the RS815, suggesting that lambda 4 could be the causal variant. To simple test to see if lambda 4 is the causal variant or not, you could just look at your data and see how it stratifies. If your data stratifies according to the activity of interferon lambda 4, so this gives the high activity lambda 4, this gives the low activity lambda 4. In the TT variant, you do not get any lambda 4. So how your data gets distributed? For example, if lambda 4 is causal, you either get two groups, high activity as one group, and the low activity and no interferon lambda 4 activity will usually cluster as one group. And if they are statistically significant, it is likely that lambda 4 is the causal mechanism. In our data, we could see clearly three groups which were statistically significant, suggesting that lambda 4 is the causal variant. A recent study used the same method and showed that it is lambda 3, because RS860 here is connected to lambda 3 expression. That showed significance, but the variants that change the function of interferon lambda 4, that is the proline and serine 70 variants, they were the same. That means lambda 4 is not functioning here, it is lambda 3. Because of the close linkage, you could use this method to come to the conclusion which among lambda 3 or lambda 4 is the causal variant. Now, completely shifting gears, I will move towards another disease called as asthma. So you all know asthma is a chronic disease of the respiratory tract. Now, what does it have to do with interferons? Because interferons are antiviral molecules. But now it may not be a surprise, because we all know that respiratory tract is exposed to all kinds of viruses, and asthma is a disease of the respiratory tract, and therefore they are closely linked. So in the epithelium, when they get virus infection, there are two lines of defense. The first line comes from the type 3 interferons. Type 3 interferons are actually called the guardians of the epithelium. They are called the guardians because they try to get rid of the virus infection without causing too much damage to the respiratory epithelium itself. Whereas if some reason this fails, then the type 1 interferons come into the picture, which are more stronger and which show much larger systemic effect. They do get rid of the virus, but they end up causing more damage to the host itself. And therefore, there was a group called the Predicta Consortium in Europe, which was working to understand asthma in connection to virus infection. So asthma is a Th2 disease. So if you know, in adaptive immunity, there are at least two kinds of T helper responses. Th1, which is good for antiviral immunity and cancer. Th2, which is not good for antiviral and cancer, but causes an allergic reaction. It is good against parasitic diseases, mainly helminth infections. So there is a connection between virus infection and asthma, because asthma is mainly a Th2 predominant disease. So if you see in asthmatic individuals, mostly allergic asthma, you see a predominant Th2 kind of inflammation that is the main culprit. And therefore, this consortium was working on that. So I went to one of the members of this consortium in Poland, to Professor Marek Kowalski's lab at the Medical University of Łódź, and where I tested whether this type 3 interferon lambda 4 has something to do with asthma. So asthma is a complex disease, as I said, characterized by variable airway obstruction, bronchial hyperresponsiveness because you are allergic, and chronic inflammation of the airways. Asthma is one of the most heterogeneous diseases you can come across. It is characteristic, which means that one person's asthma is not exactly same as another person's, and therefore his pathology is different. Therefore, his treatment could also be different. Asthma could be classified based on triggers, whether it is coming from viral, allergic, or drug-induced or exercise-induced, etc. Based on clinical presentation, like in children, it is called pre-asthma, wheezing, or childhood onset, adult onset, etc. Based on pathology, it could be eosinophilic, neutrophilic, etc. But broadly, you can classify asthmatics as atopics or non-atopics, meaning is it allergic asthma or non-allergic asthma. Non-allergic asthma could also be overlapping with another disease called COPD, chronic obstructive pulmonary disease. So doctors very diligently work to differentiate between these conditions because the treatment for them is also different. Now, atopy or allergy can be measured by what is called as a skin prick test. So here you take what are all the allergens present in your area and make a preparation out of them and just inject on the forearm and see how your body reacts to that. So if we have IgE antibodies, which are important in the, as I told, Th2 immunity means which will have higher IgE antibodies. If you have more IgE, that means you are allergic to those allergens. Immediately you see a big reaction in the form of what is called as a wheel happening. The diameter of the wheel will say whether you are positive or negative, which is a plus or a minus result for the test. Now, IgE and skin test, if you do across different ages, you see an interesting correlation. IgE is at its peak in before puberty, and toward after puberty, the IgE levels goes down. That means we are more allergic when we are reaching puberty, and as we old, we become less allergic. So basically, the goal of the study was to see whether the RS815, which generates interferon lambda 4, associates with either atopy or asthma, because all asthmatics are not atopic. I told you, some can have atopy or allergy, some cannot, some need not. So I was provided with a large number of well-characterized adult bronchial asthmatic patients, 326, and about 111 healthy controls. And the atopy data was also available for, I think, 290 of them combined, 3 asthmatics as well as controls, because controls can also be allergic, non-allergic. So, and then we diagnosed asthma based on standard criteria, and the polymorphism was genotyped. And to my disappointment, when we did an association test, we did not see any association with either asthma or atopy, because you see the p-value is not below 0.05, which is of significance. So they're all above 0.05 and therefore there is no association. But in our data, we were seeing some kind of an interaction, meaning there is some hidden interaction between the polymorphism, age of the patient, gender of the patient, and atopy. So in that case, if your statistical test confirms that there is an interaction, you could do what is called as the stratification. You could stratify your data based on what you see as interaction. So here we saw gender and age, therefore we separated our data into men, women, and ages above 50 years, below 50 years, because we thought hormones might be playing the role, and 50 years is the age of menopause on an average, and therefore we took 50 years. And now interestingly, we see that women, this is what is called as a forest plot I'm showing here. So this is so it could be below 1, the estimate could be below 1 or above 1. Below 1 means it is a negative effect. In other words, here we are testing for atopy. So if it is below 1, the chance of you having atopy is less. If it is above 1, chance is more. So as you see here, if you are a woman and if you are more than 50 years, the chance of you being atopic is less because it is lesser than 1, and it is statistically significant because it is less than 0.05. Right? So we found this interesting discovery, we made this interesting discovery that older women, if they have an allele to generate lambda-4, they will have less allergy. Now remember, this is not asthma. So we went back to those women where we had the atopic data. In those women less than 50 years where there was atopy data available. And when we went to their asthma clinical characteristics, interestingly, we saw that all these women who showed lesser allergy even if they had interferon lambda-4 generating allele also had less severe asthma in them. That means they had protection from atopy and their severe asthma severity was also less. As you see, as shown here by the frequency of their consumption of corticosteroids, inhaled or oral, ICS, OCS, was lesser, and the positive reversibility test, which is a test to test how much of smooth muscle is remaining in your respiratory tract. They give you a drug and they measure certain characteristics of how you breathe, and if you show a positive test, that means much of your smooth muscle is remaining, you have a less severe asthma. So these women who had at least 1 copy of lambda-4 generating allele, they were showing a better positive reversibility test, suggesting that asthma disease was also less severe in these women. They were, number 1, protected from atopy. They had less chance of atopy. Number 2, if they had asthma, their asthma was less severe compared to people who did not, compared to women who did not have interferon lambda-4 generating allele. Now, interferon lambda-4, interestingly, as I said, has been associated with a number of conditions. But usually, interferon lambda-4 is genetically seen to be associated with a negative result as far as virus infections are concerned, and also cancer. Because I told you, Th1 immunity is the one that protects you from both viruses as well as cancer. So people carrying interferon lambda-4 gene tend to be less protected for viruses. This was mainly seen in children, also in malaria, and also in prostate cancer. Now, so in that case, why is interferon with such negative characteristics still remaining in the human population? For example, if in India, our estimates say that more than 50% of us will be carrying at least 1 copy of this interferon lambda-4 gene and therefore protein. So why are we carrying? In that case, are we at a disadvantage to fight infections? Probably yes, as far as virus infections and cancer are concerned. But on the other hand, there is some positive side to it, because the same variant is also associated with less inflammation in other conditions. For example, if you look at liver damage in what is called as non-alcoholic fatty liver disease, if you have interferon lambda-4, you are having less inflammation compared to people who do not have lambda-4. But if you get HCV, you are at a risk. But for non-alcoholic fatty liver disease, you have a protection. So this is the dual kind of double role that lambda-4 seems to be playing. And also in another virus infection, antivirus, which is actually similar to the COVID symptoms that you get, is actually you see a protective effect from lambda 4, or rather the minor alleles show a protective effect in another virus infection. Therefore, lambda 4 story is still continuing. We need to understand its biology better. So this is what I did in very recently. We published this work. We showed that if macrophages are treated with interferon lambda 4, they will show anti-inflammatory nature, because as you see here, IL-1 beta, which is pro-inflammatory cytokine, is decreased, and IL-10, which is an anti-inflammatory cytokine, is increased. And the important thing is we want to compare lambda 3 and lambda 4, because remember, genetically they are very close, we cannot differentiate. So to understand their biology, we did the same experiment and compared with lambda 3. Very clearly with IL-10, which is a very important anti-inflammatory cytokine, if the macrophages are treated with lambda 4, you have more IL-10 compared to same macrophages which were treated with lambda 3, which actually lambda 3 produces less IL-10. Therefore, there could be the protective effect that you are getting from this apparently pro-viral but anti-inflammatory cytokine, that is interferon lambda 4. We show evidence that why it is pro-viral by using what is called as a mixed lymphocyte reaction. When we make DCs, dendritic cells, in presence of lambda 3 or lambda 4, and react them with T cells, T helper cells, the DCs were treated with lambda 4, they produce more IL-4 and IL-13, which are typical Th2 cytokines, which are antiviral or at least not pro-viral. So therefore, we provide explanation of the genetic results that world over people had seen. We show functional evidence and show why lambda 4 is doing what it is doing in genetic studies. Okay, this I come to the end of the talk. I would like to acknowledge several people here. Professor Abhijit Chowdhury, who gave us the HCV samples, and several of his colleagues, and late Dr. Amarapurkar from Mumbai, who also gave us samples. Anand Bhushan, the graduate student who did most of the lambda 4 work. Manjarika Day, who contributed to the results that I showed at the end. The other people in the lab who contributed to different areas of this work. And my funding comes from Wellcome Trust DBT India Alliance, NIBMG, as well as some grants from DBT. And I would also like to thank and now remember, because Professor Kowalski just passed away 2 months ago in Poland. So I would immensely like to thank him, and wherever he is, I would like to remember him for all the help he has rendered in this project, and both as a person but as a scientist, he was fascinating, and I cherish my association with him. With this, I end my talk, and I will leave it to Priyadarshi, if he's still there, to ask and see if there are any questions. Thank you very much.
Thank you, Sreedhar, for a wonderful talk. Alpana, are there any questions?
No, not yet, but just request all the attendees to please type in your questions in the chat box, and then we can ask Dr. Sreedhar. Please type in the questions in the chat box. We'll probably wait for a couple more minutes to see if there are any questions.
Sure.
There are no questions as of now. So I think if the audience, if the attendees think of something that they'd like to ask Dr. Sreedhar, they can always mail him. His mail ID is available on our website. So I think with this, I'll thank Dr. Sreedhar and Dr. Priyadarshi to join in for this seminar, and all the attendees for joining in. We'll have a next set of Science Setu seminar next month. So, and the information will be circulated in time for you to join in. Thank you very much.
Thank you, Alpana. Thank you, Priyadarshi.
Thank you. Thank you.